Good afternoon. Welcome back to DemoCamp. My name is Jason Best. I'll be the moderator of this session along with Deb Paul and Erica Krimmel. I say welcome back. Some of you may be new to this second session. We had 4 presenters earlier. We'll have 5 plus some discussion time in this session. DemoCamp is a format made for live presentation of software, so we can show off some of the technology, but also see it in a real-world live experience. So you get to see it, warts and all, and you might see a little hiccups, but we'll be generous about forgiving that and realizing that not everything goes right all the time. So it is more difficult than a slide. A little housekeeping. This is the second session of DemoCamp, Demo01. This year's meeting was made possible in part by our sponsors and their generosity, especially our diamond sponsors. And this session will be recorded for later viewing. We also want to give thanks to Okie Congress and the technical support here that have made the live interaction and recordings available and possible. And for our presenters, please speak slowly and carefully so everyone in the recording can be recorded clearly. And we're going to give each presenter 15 minutes. There'll be time for 13 minutes of presentation, leaving 2 minutes for questions. If they run out of time, we'll defer the discussion to the tail end of the session. We will, let's see, let me make sure I'm covering everything. If you're attending virtually, please add any questions or comments in the Zoom chat. And we will have someone locally repeat that question on your behalf, and we'll relay that information back to you. I believe that's everything for now. So I would like to invite our first speaker, Gary Motz, to present Lux.
Thanks everyone for being here today. Just wanted to give a brief introduction and overview to a relatively new service launched at Yale University that involves the collections of the Yale Peabody Museum aggregated in a single format using linked open data and a knowledge graph to expose all of the collections data information from the Yale Peabody Museum alongside all of our information from the collections information systems of the Yale University Art Gallery, the Yale Center for British Art, the Yale University Libraries, the archives, and then a handful of other collections coming along. And so we're adding more and more collections and depositing all of that in a sort of a concerted effort to sort of show off what everything has and holds for sort of an open access mandate for the rest of the world. So the URL is lux.collections.yale.edu. And just here's a couple sample collections that exist in on the site, a little bit of overview. But really, here's the magic here. And so we've got basically, when we take all of those objects and all of those works and expose them in a single knowledge graph, the back end for this really shows off just how many things are held within all of Yale's collections. So we've got more than 17 million objects, 13 million works. And so really, what's the difference between an object and a work? And you're really trying to think about the difference from like the art world. An object is the real world object. It's the thing. It's the tangible material space that occupies space. And so a painting, there's an image on canvas in a frame behind glazing with a mat and other sorts of things. So that is the physical object. The work is the image itself. It's a concept, it's an abstraction, it's something that can be represented, but isn't necessarily the paint or the pastel or the sculpture or whatever that medium might be. And so for the Peabody Museum, everything that we've got is classified as an object, but we have some works too. So when we look at all of this and take an understanding of how we can expose and find things that are interrogated in this sort of linked open data framework, we start to look at some of the ways in which we can do that. Because when you look for one thing, you get a whole bunch of other things for free. So I'm going to search for one of our more popular agents. And so as a person, O.C. Marsh is someone that was a seminal paleontologist early in the sort of beginning days of the dinosaur fossil feud between E.D. Cope and O.C. Marsh. Shows up as one of the books in the Yale University Library. And so there's a whole bunch of information that comes up. And so you can see that there are over 2,000 objects somehow related to O.C. Marsh, Othniel Charles Marsh. So we can see, oh, that's a person. there's the big guy himself. There's his dates of life and death. And so when we pull this up, we just get this agent page for O.C. Marsh. And so this particular individual has a whole bunch of information because we have information about objects that were created, encountered, or influenced by them. So helpful language for talking about both the art world as well as natural history and cultural heritage situations, but also works created by. So, so when we get to the point where we're looking for other things that Marsh has influenced, we've got publications written by him or about him over here on the third tab. And so we've got more information about how O.C. Marsh is really represented in all of the collections across Yale University. So if we want to take a look at other things that we might want to see, let's look to search for like agates. So agates are really well described in the Peabody collections, and so we've got some nine-eyed agates, we've got the cutting agate in the library. Let's look where things have digital images. Okay, we've got some beads, we've got some medallions. Let's make sure that we're doing this from the Peabody Museum, just because those are the ones we care about. Oh, we've got some more beads from the anthropology. I was really interested more in meteorology and meteoritics. I'm looking for those actual minerals. Oh, quartz, var, agate. Okay, so we got some more information there that comes up on a particular object. And so if you click on an individual record, say, oh, this is a nice, pretty specimen that renders in a IIIF universal viewer, so we can view more information about that particular specimen there. So agates are good things to look for, and so a number of people have talked about, so what if I've got a preparation or a part or a component, or how do I represent an object in the data that is a skull or a part of a skeletal element? And so if we're looking for the skulls that might exist in the Peabody's collections, let's go back down and make sure that we've got the responsible unit, also the Peabody Museum. And so we see a whole bunch of things like, oh, Rhamphorhynchus chillingi. Here's a cast from Vert Paleo. There's a whole bunch of things in here. Oh, there's an image. Here's another one. Here's another. So we've got a bunch of different images and views from multiple objects within this lot that Vanessa has put together and is now exposed on the collection object. And so we can see that this is a physical object in the Yale Peabody Museum's collections. And so when we're really trying to expose the accessibility of these objects, we're not just doing it for the information that we have about the metadata, the digital assets that go along with them. We're also trying to say, oh, how do I see it? Well, it's in the collection. Let's take a look on the Yale Peabody website or contact Vanessa for more information. Where can you find it at Yale? And so this will direct you to the collections manager's contact information. You get a little bit more information and a scalable map about where it was found and where we can go with that. So let's do a couple other searches here and see what we can find. Simple searches give you one thing. So if you're interested in dire wolves, you can come up with a number of results, and so you get 13 objects. But let's see if we can do a little bit more of an advanced search and show what you can do here. So with an advanced search in sort of knowledge graph expression, we want to search for a couple of different things. And so just for ease of access here and to sort of guide those that might be interested in sort of how to get familiar with searching a knowledge graph or some of this, we've got some helpful hints here and additional advanced search examples. So we're going to come in here and look for dire wolf specimens found in California. And I'm going to cheat a little bit because that puts California in 3 different places. So if I'm really interested in those dire wolves from California, we need to know whether it's found in a place called California, mentions anywhere in the metadata about that object something that is called California or is associated or reconciled to that concept that is California. So it gives us all of the information at a whole bunch of different levels, so it gives us some specificity to make sure that way if there is any variation in the way in which those data are represented in our collections management system and then represented in this discovery site, we're able to sort of help to overcome the data quality inconsistencies that may exist and put those out there. So I'm going to take one more look and say, oh, let's look for my favorite taxonomic name on planet and see what we can come up with here. All right, not a ton, but all right, let's see. We got there. Got a couple of images loading, and got some physical objects. Other identifications. Ooh, Smilodon californicus. Well, there's a bunch of other information. I'm gonna go back because that didn't give me exactly. Oh no, what we're looking for. But ultimately, what we're interested in is how can we expose all of this information and make sure that we're able to see what the collections discovery site can do for you.
All right, thank you for proving the possibility of what can go wrong. We'll see if this works.
Thanks for the assist, Jason.
Let's see. I think they're doing
It's got the extended series.
it. Yeah. Come on.
Anyway, while Jason's getting my backup option ready to go here, this was a live demonstration. These are always fun, so I was doing some planning earlier this morning. But there's some postcards up here with a little bit more information about where to get this, and we're also making sure that this kind of linked open data discovery layer that aggregates information about all of the collections across Yale University's collections is something that is accessible to everyone across the world. And so we want to make sure that the architecture, the framework, the data processing, the reconciliation pathways that we do with all this, all of the information that we're getting from here. So if we look for Smilodon fatalis real fast. Let's come over here to concepts. So concepts can be done a little bit differently for art concepts as well as for taxonomic concepts. So when we pull this up here, we can see that other taxonomic concepts are linked in and exposed as well. So there are synonyms and homonyms and all sorts of other taxonomic related searches that can be brought up that are reconciled in the back end because of fuzzy matching and a couple other identifications that we do to expose the data via linked data. And all of this work that we're talking about here is not something that's done in the collections management system. This is done behind the scenes in using Python scripts that go out and reconcile against Wikidata Q numbers. So we can say this taxonomic concept is the same thing as something over here. And then when we combine them together, we get more information that's federated into this one-stop shop. So when we get the Felis fatalis, Smilodon fatalis, the common names, the general species concepts, and then hierarchies that include lots of non-Linnaean taxa as well, that's not something that we record in our CMS. That's being added from external sources. So when we pull up that particular record and you really want to see, oh, where can I learn more information about where that came from? That is not just coming from Yale. There are information about the JSON manifest for how that 3D or how that image gets represented, where those image, the metadata for that object come from, and then also contributing records that may be from Yale Peabody Museum, another Yale institution, or from external sources like VF or LCNAF or Wikidata and all sorts of other information sources that are exposed data and information in linked open data. So there's a whole bunch of stuff out there, and I apologize, I blanked out on some more tours, but effectively what I'd really like to make sure that you know is that if you are interested in taking a little bit more of a look at where the source code is, the GitHub is here in Project Lux, and so we have our front end, back end, middleware tiers, and the data processing pipeline are all in public Git repos for availability, so that you can take a look at how we're reconciling information from all of our internal sources and sending that out to other external data sources as well. Thank you for your attention, and thank you for your computer, Jason.
We have time for one question. If anyone has any questions, do we have any online? Okay, thank you for sharing that code. I think a lot of us will want to look into that. Up next, we have I'll mention it from up here. Okay, up next, we have a presentation. It is not live, but live recorded. It is a video, but it is in the spirit of DemoCamp, recorded in a live demonstration. And I'm going to ask James Mickley to come up and introduce our next presenter.
So I am not the next presenter. It's Will Weaver, the creator of VoucherVision. And so Will is going to show us this demonstration, and it's 3 in the morning where he is, but he's on Zoom, and so he can answer questions by the chat and hopefully live after his video. And I just want to say, I think that VoucherVision is a transformational tool. I hope you all try it. And I hope that we, as a community, can find ways to support this work, because I think it's really helpful. So, is there a way to start the video?
Hello everyone, my name is Will Weaver and I'm a PhD candidate at the University of Michigan, and today I will be demonstrating our museum specimen transcription tool that we call VoucherVision. And first I just wanted to thank James Mickley and Jason Best for helping get this all set up. It was a little chaotic since I'm not going to be there in person. But anyway, so VoucherVision is one of a handful of AI and large language model powered transcription tools that folks are developing. And our focus with VoucherVision is to create a tool that's simple for most herbaria to use, that keeps all of the transcription processing in-house, and something that's easy enough for individual researchers to use as well. And our goal is basically to not require herbaria to change their existing procedures all that much. Basically, we just transition folks who would otherwise be doing manual transcription into editing the large language model generated transcriptions. So in that vein, let's get to the demonstration. VoucherVision is split into 2 parts. We have the actual transcription tool, and we also have the editing tool. And we'll start by taking a look at the actual transcription tool. We made a graphical user interface just to make it easier for folks to use, but you can also just run this using a configuration file from the command line if you wanted to run it on a cluster or something like that. So the first thing we could take a look at is our prompt builder. The version that we're hosting right now is based on prompt engineering. So we have to give the large language models a set of instructions, and that's how we get an output. So if you open this, it'll let you start with the template that we use for VoucherVision, but we could load in some other existing prompts and take a look at what is going on there. So basically we have a chunk of instructions, and then we have individual fields where we give it a pretty lengthy description of what we want it to actually transcribe. Through some empirical testing, we've observed that large language models can handle and perform better with complex and detailed instructions, whereas some smaller models, like a lot of the open source models, they've been working better with shorter prompts. So here you can see that we have a bunch of different prompts we can play around with. We've also been working with some other folks to create prompts for things that are not herbarium specimens. So some folks at the Field Museum have been helping us expand this into mammal specimen tags. So you can see here that a lot of the fields are the same, but as we go down, let's see where they are, we start having measurements that you wouldn't find on an herbarium specimen, for example. All right, so now we can head back to our app and take a look at what we can do. Moving down, we can also pick the version of the large language model that we'd like to use. We'll come back to that in a minute. And then we also have different OCR engines that we support. So right now we support, it looks like 5 different OCR engines. We have everybody's favorite Google Vision API, but we also support Lava, which is a vision language model. And we support the Craft plus Transformer OCR model. And then we can also use GPT-4o Mini as the actual OCR engine. You could also do that for GPT-4o itself, but GPT-4o Mini is much cheaper at the moment. So our favorite though right now is Florence 2. It can run locally and it's really fast. So that's a really good choice for us. So if we wanted to work with both though, the way you set up voucher vision is you can have multiple OCR engines feed into the large language model at any given time. So we can use both Florence 2 and Google Vision to help our models get more information. So once we pick our OCR engine, one other thing we need to set is basically what kind of pre-processing we want to use. So if we take a look at an example of a label collage image, we can use Leaf Machine 2 to crop out the text of the image and get a label collage, which simplifies what the OCR engine has to look at. We can also do something pretty interesting with the mammal specimen tags. They've organized them, they have images that can be parsed based on their file name, so we can convert them a little bit differently as well. So anyway, so we'll start off by using the original image for this test, just for the sake of time. And then we can just use our basic defaults. We found that Google Gemini 1.5 Flash is a really good base model for most scenarios. So that's it. So then we can hit start. And if we go take a look at our terminal, we can see that in the background, behind the scenes, a lot of other things are going on. Right now, we're just verifying that the images are all valid and making sure that the orientation is correct. And now it's loading our OCR models. So it's currently sending a call to Google Vision API, but it's also loading the Florence 2 OCR model at the same time. And the first time you run that, it takes a little bit longer, but once it's loaded into the GPU memory, Florence 2 runs pretty quickly. And we've also found that with Florence 2, it loves to not add spaces after periods sometimes. So right now with Florence 2, we're actually passing the output of Florence 2 to a Mistral quantized model so that we can fix the Florence 2 formatting mistakes. But I have a feeling that Microsoft will end up fixing that later on, so we don't have to do both at the same time. We can also look at our GPU utilization. And like I said, because we're loading Florence 2 and a Mistral model, we are using a fair bit of GPU resources. But if we didn't have to use Mistral, we would only be using about 12 gigabytes of VRAM for that. So now you can see that our OCR finished. And if we look up here, we have OCR from Florence 2, and then we have OCR from Google. And then we finally have our output model. So if we take a look back at our graphical user interface, you can see that it looks prettier. And basically what we have going on here is when Voucher Vision runs, we also send queries to third-party APIs. So we're also querying WorldFlora Online and a geolocation app to try to fill in extra information. It's pretty common for some information on labels to be incomplete with reference to geography. So we found it's pretty useful to get things like continent or country in a more consistent format using this. And then just for a sanity check, we can see on the map where these specimens are, just to make sure everything is working as we would expect. So that's cool. And now if we wanted to run something entirely locally, we do support quite a few different models with Voucher Vision right now. We support all of the OpenAI models. We support using OpenAI Azure endpoints, and that's basically for the University of Michigan. We have our own version of OpenAI. And we support several Google versions. Right now, Gemini 1.5 Flash has proven to be really cheap and also really effective. And we support all of the API Mistral models. And more importantly, though, we have quite a bit of support for locally hosted models. So you can see here, we have some fine-tuned models that we could run, which we've trained on synthetic OCR data. And then we also have some CPU-only models. These are useful if your institution already has access to some computers that might otherwise be used for genomics, something with a lot of CPU horsepower. And then we have some other basic Mistral models. So right now we don't have time to go into any more of those, but you can play around with that, and then you can see that we have quite a few local models that we have access to over here. Okay, so moving along, we can take a quick look at our Voucher Vision editor. Basically, the output from VoucherVision, you get a zipped file, and all we're doing is providing that to the VoucherVision editor. Once we load that, we can pick a transcription sheet and start editing it. And right when it opens, you get some user interface selections. So for example, if we wanted this image to show up smaller because our monitor is smaller, we could change the size of different components of the actual GUI. You can change width, things like that. Make the image bigger. We won't deal with that right now. So anyway, what's going on here? This is the actual record that we received from the large language model. And on the left, you'll see any of the hints from the geolocation tool or the World Flora Online, if they happen to be present. So we can review all of the inputs, make sure that they're correct, and we can confirm that and move along. And now you'll see that we have some geolocation hints that are showing up. We also have a map over here, so we can see if any of the GPS coordinates are correct. So you see a blue dot in Nevada. If we wanted to zoom in and see some really fine detail, we can open this in a new window. We can also take a look at the Leaf Machine 2 label collage and see something like this. And finally, if you use the Google Vision API, you can see the OCR quality scores, see how accurate the OCR is. We also automatically include some links to really helpful sources, including Plants of the World Online. And then we also have links to the USDA. You can get more information from that. And then finally, we also have VoucherVision running on a Hugging Face space. So this only has access to API models. So if we want to hide this real quick, we can start fresh. Basically, you could upload images here. I guess one other fun thing I could mention is that we've been working with USDA Wood Laboratory. So we've added support for PDFs. They have a lot of wood collection PDFs. So in this case, we can upload a PDF, convert the pages into JPEGs. And now we have 50 different index cards from a wood specimen collection that we could process too. So that's fun. And then, like I said, on this Hugging Face space, you're welcome to go there and play around with it if you want. Some of the models won't work. So like right now, we can see that we don't have access to any local models because our server doesn't have access to a GPU right now. But if you wanted to pay a little bit extra on Hugging Face space, you could pay to have access to a GPU and run local models on the Hugging Face space if you wanted to. We don't have access to Mistral models just because I don't have academic funding to provide that to everybody else, but we do have funding to give people access to use any of the Google models and also the OpenAI Azure models through the University of Michigan. So yeah, you can play around with that. You can build prompts yourself and then test out a bunch of different things. Great. Yeah, so I hope James can answer some questions for you. It's 3 a.m. where I am by the time we're presenting live. So hopefully James can answer some questions. I'll try to be in chat, but I can't promise anything. So thank you for your time.
Okay, we do have time for one or two questions, and we can play Stump James, or maybe we'll just talk about LLM. Oh, is he online? Okay, I didn't see him in the Zoom, but okay, so are there any online questions? Okay. Any local questions for Will? He stayed up very late for this. All right, I will say that I saw that there was a calculator for how much it costs to run. I think it was 7 cents or something, so that was a pretty cool feature. But I don't know if that's really a question for Will, but I like that, Will. Yeah, we have a question here.
Hello. I just have a question about the PDF one. So that was really, really cool. At the moment, I am just cheating and turning PDFs into images myself and then using LLMs, whatever. And the reason why I've done that is because sometimes they are rotated weirdly, certain pages. And I was wondering when you are uploading a PDF, if things were rotated incorrectly or rotated differently across pages, does VoucherVision automatically fix that? Or is that too complicated? And did my question make sense?
Does my microphone work?
Yes.
Great. Okay. The short answer is if you know that it's going to make a mistake, we have some buttons you can select to automatically rotate things. We're also just converting it back to images right now, not dealing with the PDF directly. Some of the OCR engines don't care what orientation things are, like Google Vision's API is really good at reading text in any orientation. Some of the vision language models do struggle with rotated text, especially if it's mixed text. So if some of it is horizontal and some is vertical, you might get mixed results. Google Vision, though, seems to be good for that, which is why we also support sending multiple OCR engine text into the large language model. So theoretically, you could take advantage of both.
Thank you.
Thank you, Will, and thank you for staying up late to entertain our questions. Our next presenter is Lindsay Powers, and she'll be presenting fair data, I think.
Hi, all. Thank you for allowing me to talk today. My name is Lindsay Powers. I'm a program director at the U.S. Geological Survey. I am not a technologist, but I'm going to tell you a little bit about some tools that we've been working on and working with to try to create a collections management system for USGS, which has broad responsibility for managing a lot of different types of scientific collections, but we do not have a museum mission. So we're a research organization trying to get a handle on what we have in our broad collections across our organization to make them publicly available, discoverable, and reusable. My program, the National Geological and Geophysical Data Preservation Program, has a mandate from US Congress to create a national catalog of geological and geophysical resources that our program funds to be preserved and made available. So we fund primarily state geological surveys and USGS scientists to take historical data, samples, maps, well logs, anything that's related to the geological or geophysical research enterprise, preserve those to modern standards and then make them available. So this tool was originally developed to catalog these resources that we've funded through the program, but now we're trying to expand this resource to serve USGS as a whole and also the state geological surveys. So we're going beyond the bounds of just our organization to try to serve the needs of a really broad set of stakeholders for a lot of different types of collections. Let's see. So also this work is in progress. Hopefully it will work today, but it's in kind of ongoing development. Here it says that the recycle the registry of scientific collections. So it's a little bit of a play on words because really the focus here is we want our collections to be reused by people. So we have this long history of using this, the previous National Digital Catalog as this national catalog. It's been re-architected. So these 3.5 million items that it says we have, unfortunately, those are not very well described in this ecosystem currently. So we're focusing on really upgrading and updating metadata to describe these resources. So the tool itself consists of a searchable map, an inventory that's organized by organizations, and then this collections management function that in this tool, we're really focused on describing things at the collections level, but we have the capability to add items into those collections. So if we just look at the map function, so here you can see that the collections themselves are grouped in the map based on where the collections currently, the geologic or the geospatial information indicates that they live, which in many cases currently is at the site of the State Geological Survey. So we're working to try to update the collection metadata so that it's actually reflecting where the collection itself came from, but that is in progress. But I just wanted to show we have a collection sitting out here in the Ryukyu Islands here. And so if you use the map function, you can click on the collection itself and you can see the description, the general description of the collection. And then you can click further into the more robust and rich description of the collections, which includes here the location on a map interface with the bounding box, tells you how you can find the collection itself, and then details about the collection and details about the metadata, who created the metadata, and the history of the metadata itself. We use controlled vocabularies to provide keywords that are describing the collections, and the controlled vocabularies that have been used to create these keywords are listed here with the keywords that are selected. And then if there were items associated with the collection, they would be listed here and potentially be linkable or have images, but you'd be able to find out where you could get access, what the access restrictions are, et cetera, for the collection or for the items themselves. So in describing the collections at the collection level, we've come up with about 15 required metadata elements to describe the collections. And then we have a lot of recommended options, and I'm going to talk a little bit about how you create the metadata, how we create the metadata records for these collections. And then we also have required and recommended attributes for the item level descriptions. And again, here we're talking about anything from eDNA samples up to, volcanic rock and kind of everything in between. So trying to be as encompassing as we can, but also trying to get people to provide as rich metadata as we possibly can. So the tool that we use, the tools that we use to create the metadata records are within the MD Toolkit, which is a suite of tools that was developed in a collaboration between the U.S. Geological Survey and the U.S. Fish and Wildlife Service through the Alaska Data Integration Working Group. This toolkit was established quite a few years ago, but it's an open source suite of tools that we wanted to incorporate because they have a lot of flexibility. They allow us and others to create specific profiles that can be incorporated in the metadata records. So you can control, what people can enter and or people can select to have full profiles where they can have access to anything. The other thing that is nice about this toolkit is that it has a schema tool function. So the MD editor, which is the editing part of the toolkit, it outputs in several different formats. So it writes an MD JSON and then translates to ISO, HTML, MD JSON, and a USGS metadata standard that's not widely used internationally anymore. So we have this translating function at the back end of the MD editor, and I'm just going to pop into the MD editor itself. So the way that it works is it's a web-based browser tool. So you're creating this record in your browser. It's cached, and that can be a problem, so you have to beware to save and save often. But it's cached in your browser, and then you export it as that MD JSON or some other you can use it for anything, but for us, we're exporting an MD-JSON and then ingesting that into Recycle as a collection record. So up here, you can see the profile function. We have established 5 different profiles that are specific for our program that help guide people to the types of metadata and also the controlled vocabularies associated with the attributes that we're asking for in these profiles. So here, for this particular record, we're using the geological profile because this is a collection of halite cores from New Mexico. Over here, you can see the contacts listed. You can manually enter the contacts here, or you can import them in a CSV file. And then those contacts can be reused over and over again to create collection records. So this first page, the main page, has kind of the general information about the collection, the title, et cetera, the contacts. The metadata tab is metadata that is specific to the metadata. And then the keywords tab is where I think it gets interesting in that you can add thesauri or controlled vocabularies to add keywords. And so I don't remember how many controlled vocabularies we have in here, quite a few. Many of them are hierarchical, and they're geared towards geological collections in this case. We also have an invertebrate and a vertebrate collection that we're working on, and we're adding controlled vocabularies to those profiles that will be specific to those types of collections. So we encourage people to add as many keywords as possible. Here is the geographic extent of the collection. There's a section that talks about the lineage of both the collection and the metadata, and then constraints on distribution, how you can get access to the collection, and use constraints. And then you can add associated resources. And the terminology here is not what we would have chosen exactly. It can be a little bit confusing, but we are kind of bound by using this open source tool that has been developed by other people, so we're trying not to change too much. So anyway, you can include associated documents, and then we also have the ability through the Recycle tool to issue unique identifiers, both at the collection level and at the individual item or specimen level. So we're issuing DOIs for collections as their collections of metadata or data. And then at the individual sample level, we are encouraging people to use international generic sample numbers so that they can be tracked and tied to all of the associated resources. That's it.
Okay, thank you, Lindsay. I think we have time for 1 or 2 questions. Yep. One coming from the back. Are there any online that we need to be ready for?
Thank you very much for your presentation. I was wondering if the data that you hold can be accessed programmatically. Okay, so I work on the Global Registry of Scientific Collections, and it looks to me like you have amazing data. It would be nice if we could use you as source of information
Yes.
for GR Cycle, like we do for Indexa Bioram, but it requires the data to be available programmatically. So
Yep.
that was my question.
Yes, our intention, because we are a federal agency, so we, I guess, are somewhat bound to provide our data to external organizations, and certainly GR Cycle is our end goal. So we will be developing the ability to push or to have you pull from
That would be
from our system.
that would be so great. So we'll be in touch.
All
Thank
right.
you.
Any other questions for Lindsay? I don't think so. All right, thank you. Next up, we have Shelley Gaynor to present some R code data cleaning.
Hi, I'm Shelley, and today I'll be presenting on an R package called GATORS, which stands for Geographic and Taxonomic Occurrence R-Based Scrubbing. This is a package that was developed by then undergrad Natalie Patton. She's now a PhD student at the University of Ohio, University of Ohio. And everything that I'll be showing you today, though it's written in R, you can see it on GitHub under mgaynor1/demogaynor2024. This is going to be shifting up what we've been talking about. A lot of the presentations today have focused on, creating the specimen record or, updating that specimen record. This is taking the view of a researcher. How can we get the records we need to understand species? And so my focus really was how many digitized records exist for the Southern Appalachian endemic Gaelic Ceciolata, which is in the family Diapensiaceae. And so because none of you probably know what that is, I'm going to introduce Gaelics and then demonstrate the many, many ways to download it, including in our GBIF, our IDIC-Bio, SPOC, and with our package GATORS. And GATORS doesn't just download occurrence records, so I'll introduce you to some of the features, but not all of them today. So Gaelic Ceciolata is in the family Diapensiaceae, which most people do not know, even if you're a botanist. This is in the order Ericales, so think blueberries or tea. And some of the species I've listed up here, but I will not try to pronounce these names. Just in case, from whatever country you may be from, these could be very well known. But Gaelics has these beautiful leaves that people poach for floral arrangements. It's called a wand flower, which you can see from its wand, and it covers the understory. Like I mentioned, this is a Southern Appalachian endemic, so here we're looking at the eastern US, and it ranges from Alabama all the way up to the northeast. And there's a lot of taxonomic issues. And so all of this is on the Wikipedia page for Gaelic, so if you get lost here and you're really interested and want to know more, please go to that Wikipedia page. But it was identified in the 1730s by the first John of this story, John Clayton, and it was named Gaelics Aphylla. Now, he sent this to Gronovirus, who published it with a different name, but all these specimens were destroyed in a series of unfortunate events, including the World War II bombings. However, zoom backwards to before all these specimens were destroyed, a different John, John Mitchell, collected what he thought was Gaelics. However, it was Nymphalia aphylla. It's in the Boraginaceae. It's not Gaelics. You look at it, it's not anything like it, but it was mistakenly labeled as Gaelics aphylla, and on the way, all the specimens were destroyed by pirates, and so Linnaeus never got to see it. These pirates took it away from him, and even so, Linnaeus accepted this designation, and this has been horrible for all of our specimen records, even today. This is an issue that still persists. This is a monotypic genus. This is the only species. And still, this issue, this acceptance of pirate-stolen specimen, led to a lot of unfortunate events. Also, because, this is back in the 1800s, between March 1803 and 1804, it was renamed 5 times. And all of these names that referred to the Linnaeus specimen were then invalidated. So the only name that was actually okay was based on a plant in cultivation in France, and that was Pyrola uceolata, but this never caught on. Everyone knew this plant as Gaelics. They were not going to take it as Pyrola. We had to rewind, but we did get the uceolata from that. The current type specimen for Gaelic Ceciolata, since all the others were destroyed, was from Andre Mitrotz, and on the way to get the specimen, his horse were stolen. So really, this is a hard one to keep it together. And he labeled this originally Gaelics aphylla, but now that really does belong to Nemophila aphylla. And on GBIF, when I look through all these specimens, there is only one that is labeled Gaelics aphylla that doesn't belong to Gaelics uceolata, and plot twist, this doesn't belong to Nemophila either. So we have all of these that are currently being renamed, but they belong to Galexophilia or Galex uceolata. They're currently all assigned to Nemophila aphilia. So if you work on Nemophila, please keep this in mind. But I'm currently fixing this by going to the actual specimen and relabeling them because there's no other way to fix it. Anyways, keeping this all in mind with this monotypic, very, very simple genus, very simple species, this could occur in any species that you're interested in working on. Doesn't matter what it is. Story might not be as fun as Galex, but once you suddenly dig into it, you're going to find that you have to know the synonyms that belong to your specimen in order to actually collect what you think you need to understand your species. So now when we're digging into how we actually download this, I'm not going to actually run these live. I'm sorry, but it wouldn't finish in time. APIs are quite slow. But the first way to do it is with our GBIF. And there's two options. You can do this by scientific name, and it's a pseudo fuzzy match. And as I say that, please ask someone who works for GBIF to make sure that's the correct way. But what you can do here is use their function awkdata with both scientific names, Galex uceolata and Galexophilia. You can limit this to about 8,000 specimen. The reason why is this is automatically limited to about 2,000, and that is not what you need. It goes up more. Then when you actually look at how much you get, because this is downloaded in 2 separate data files, you get about 7,000 records. To make sure you know what's going on here, I'm actually going backwards and re-downloading based on the data keys. Don't worry about the details here. Basically, all I'm doing is getting the verbatim names. And again, if you want this code, it's already online, it's already on GitHub. Here you can see that these are all Galex uceolata with, the corrected names. There's no misspellings here. This looks pretty nice. This is what I think should be there. The second way to do this is through the taxonomic backbone for GBIF, which many of you probably already know of. But here you download your taxon key, and then you go backwards and do that awkdata function again with that taxon key that you identified for your species of interest. I'm only showing one here. You would have to do it for both Galex uceolata and Galexophilia, but if you do Galexophilia, you're going to get Nemophila with it because it's only seen as an accepted synonym and not a accepted taxonomic name. Moving on to our IDIC-Bio, here what I'm doing is I'm just searching using the record query with a list with scientific name equal to Galex uceolata or scientific name equal to Galexophilia. You only get about 1,692 records. And one thing you should notice here, this is big, important thing, this is an exact match. I'm only getting things that are exactly equal to the scientific name. And this is often forgotten with our IDIC-Bio because we don't function like our GBIF. Or we don't function not only in our RSense, but in the API. And so Spock, when you go through and you download, it's very simple. You could do awk to data frame with your download doing Spock awk, and you just list your names. You want it from both of these repositories, and you end up getting exactly what you think you would get. Some things that are misspelled, that lowercase is from IDIC-Bio, but move into Gators, which really I'm jumping forward, but Gators, you list it very similar to Spock. You just list a list of synonyms and you download it. One thing you'll notice here is we actually grab some misspellings. That's because this is what the provider gave to IDIC-Bio and to GBIF, and we're not using things that have been standardized because I don't trust them, not when they're renaming Galexophilia as Nemophila. It is not a boraginaceae, and I really need my diapensiaceae here. And so the takeaway is that what you see here, the filled in color is IDIC-Bio, so it's the number of records that we got from IDIC-Bio, and the clear color is what we got from GBIF. And overall, Gators gets more records from IDIC-Bio. It's not actually getting more records from GBIF because it's not made to. It's made to actually adapt to how IDIC-Bio functions and get as many records as possible. And you might be thinking, aren't they the same? Don't they overlap completely? They actually don't. IDIC-Bio still has records in it that GBIF doesn't have. And so you really need those records. And as the study that actually publishes Gators shows, especially for rare and endangered species, you need to be using IDIC-Bio and GBIF to get all the records that encompass your species of interest, in North America at least. Now, my focus here was really just to show you our really great download feature because we spent a ton of time on it, but we also have a ton of other cleaning occurrence record features, including taxonomic harmonization with a function called taxoclean. We have locality cleaning with basic locality clean, and this really cool interactive function that Natalie spent a ton of time on called process flag, which I would show you, but I don't want to mess up my computer right now. We also have a remove duplicate function. If you want to talk about what type of duplicates, we have aggregator duplicates removed, as well as occurrence ID based duplicates, so things that were duplicated due to the collector. Because this was focused on things that we do in our lab, in the Soltis lab at the University of Florida, we also have things like basics cleaning and thinning points and 1 point per pixel, because we do a lot of ecological niche modeling, and this is just part of our process. And of course, because we don't ever want to forget the collections, we have things like citation bellow, because a gator bellows. We also have remove redacted. So if we contact a collection and they give us redacted specimen records, we have a function so we don't end up accidentally publishing that with our data. We also have something called data chomp, which just gets our data ready for some other downstream applications. Here I show this taxonomic harmonization, where really what it does, in our case, we accept all these names. This is one of the many names that occurred between 1803 and 1804, and it's an accepted name. And all of these we know belong to Gaelic Sociolata, so we don't actually need to apply this here. But if we did, what it would show you is what your name are in your data frame, and then show you like what it's actually accepting with a fuzzy match, an exact match, or an interactive match. Here we have our basic locality clean, where we remove any records at 00. It's a plant, it's not going to happen there. The lat longs are rounded, and we round them to 2 decimals, and we remove anything that's identified as skewed, so flagged for skewed. And here is just a quick map of what that looks like. And with that, that is gators. Yeah.
All right, you brought a cheering section. Are there any questions from the audience or online? No questions yet? Well, it's great to see some of the under the hood things from this presentation and voucher vision. We don't want to jump ahead too far because we'll get off schedule and someone may be jumping around. So I don't know. We talked about regex last time. I don't know how to kill time this time. We could talk about R. Has anyone used AI to generate R code? Yeah, so if someone has a question, please come forward. I don't want to waste time.
Sorry, maybe I missed some of it. You were speaking very fast, but so if I understand correctly, the package queries both IDIC-Bio and GBIF, right?
Yeah.
And I saw at the end you have this citation below. So does it mean that you, because I was looking at the functions you were using and you were doing like an occurrence search, which doesn't generate a DOI to cite. So how does it work if you don't?
So citation bellow is based on GBIF citation because IDIC-Bio doesn't have a citation function because there's not just a DOI associated. But we do keep enough information that you can actually track back which institution you're getting records from. This, however, only focuses on the citation via GBIF because citing a record that belongs to like a different organization only hosted on IDIC-Bio, there's no true feature yet.
So you don't generate a download then?
Even if you generated a download with IDIC-Bio, our DOI isn't stable.
No, I mean with GBIF.
Oh, you do?
You have a DOI.
You can get a DOI assigned via their citation function. I can send you a gist that shows you how to do this, but
no,
it's...
I mean, okay, so you're making a derived data set then?
Yeah.
Okay.
I trust Erica, yeah.
Okay. No, yeah, just, thank you. Sorry.
Any last questions? No. Okay, thank you very much, Shelley.
Am I now? Just that. Great.
A little bit ahead, but I think we'll move on. We're not too far. I guess we're ahead. So our next and final presenter is Nikki Nicholson, and that presentation will be followed by a 15-minute discussion.
Thanks, Jason. Yeah, I'm Nikki. I'm from Q. I'm going to demo a piece of software called Obsidian and some work we've done on top of that in a project called Spnhc, which is an extensible notebook for open science on specimens. So I'll do quickly some Obsidian basics to start with. So this is an Obsidian window. The data in it is all formatted in Markdown. So if I flip into the edit view, you can see that this is Markdown format text data, just like you might use if you edit a document on GitHub. And it's a really good source format. It version controls very well. You can use it to build static websites, the kind of things that we see in the GBIF hosted portal. And the data is stored in files on your file system. So this is a piece of software that is built for note-taking. And I guess a lot of people were burned by note-taking apps getting bought out and then people imposing restrictions on whether you can actually get at the data that you've put in your system. All your data here stays locally, or as local as you choose it to be. So I can see the data I've got here. I can show you it's in my file system. It's all local to me. You can also store PDFs in it. So there's a PDF viewer. So I can drop a PDF in. I can access all the data that's within that. And you can attach it to a reference manager, but I've only got a very short amount of time, so I'm probably not going to do that just in this demo. But if you want to see that, just grab me. I'm here for the rest of the week. Obsidian is all about links. So it does both simple links and transclusion. So I can make a simple link just here. If you can see where my edit cursor is, then I can link to the README that I've got in this project. If I hover over that, you can view the page that's on the other end of that link. But it's a little more advanced than the linking system that we've got on the web, because it supports this thing called transclusions, which were proposed in the very early days of the web. So now if I've put that exclamation mark in front of the link, it's going to embed that file within the file that I'm looking at the moment. So when you're building your system, your project, you can be really modular about the things that you create. And we've designed, well, our interest in this was really to support people doing taxonomic work. So people working with specimens, writing literature which cites a lot of specimens, cites a lot of existing work. They can be very modular about the notes that they make and how they compose their manuscript. You might notice on the interface there's no file bar at the top. Everything that you want to do, you get at through a command palette, and you can search that. I find that quite useful because I don't have to remember where anything is. I just have to remember what I want to do, and I can type it in and find it there. You get a graph view. This is quite an empty vault since it's a demo, so I'll probably do that at the end. And it's cheating for the DemoCamp format a bit, but you can also flip all your notes into slides. So if I do slide view, I can actually like sneakily do a presentation as well as a demo. So yeah, but I'll get rid of that. The design idea behind this package is that they provide the skeleton of the package, and other people provide plugins. So I can show you the kind of plugins that are available and the number of them. There are core plugins that the team provide that look after the software itself, and there's like a couple of dozen of them. And then there's community plugins. And if I browse those, you can see there are 1,800 plugins, and some of them have been installed like 2.5 million times. So I was quite happy to see the level of use that this gets, in that we're all busy people. We've all got too many projects that we look after. I want to use things that are quite generic and know that if anything goes wrong with them, there's a whole team of people around me, around the world, that are going to help fix it. So yeah, given that all those plugins exist, there's quite a nice community that's quite helpful, that help you know what to do if you want to develop your own plugins. So that's what we did through this project. So I can show you a couple of demonstrations of that. I can search for a name in IPNI. Actually, I've got really limited time, so I'm not going to type. I'm going to paste. Search for a name. I get a list of matches back. I get data back from the IPNI API, and I get data back. If I show you, it's in Markdown format, but it's got some structured data at the top, including the name ID. And it's got a link to the DOI in which that name was published. I can search for the type. And I can grab the data from GBIF, and I get a nice, like, zoomable specimen image here. And I can look at where a specimen might be held by looking at GR cycle. And I know that was really popular because I had to sit on the floor today in that. Where was it? There we go. Yeah, so I get some data back about Q. So in my abstract, I put in that I was going to talk about us loading contextually relevant content, and this is loading data from Wikidata. So if you were here on Monday and you saw Siobhan Leachman's talk, that was a really great evangelical talk about Wikidata. So if I go to IPNI and look for that name, what I'm going to do now is show you a sidebar, and I'm going to say, let's go to Wikidata, and I've mapped some of the front matter properties against the properties that I know exist on Wikidata. So I can load contextually relevant data from Wikidata using that command that's highlighted there. So it's taken an IPNI name ID, it's run it against Wikidata, it's brought data back from Wikidata, so it's got a Q number for that species. But the useful thing for you as a researcher is like this enormous list of links that you've got of IDs where that species exists in other resources. You don't have to manage these, but if they're things that you want to consult, then you can look at those as you're doing your work rather than jumping out to your browser all the time. That is about as far as I had got when I put the abstract in for this conference, because it was quite early in the year that we did that. But since then, we've been experimenting with connecting it to an AI co-pilot, so access to a large language model, which is also what we've heard a lot about this week. So I've got a couple of demos of that too. So I've got a set of specimens here that you might have grabbed out of a piece of literature that you might be working with. And if you look at the right hand of the screen, then I've got access to a large language model which is running as a service. And I can send this note to that service. And the robot says, yes, I've read these notes, so I can do something with them. So now I can ask it to do something. So I can say, what I want is a Dalrymple table. And I get that back. Then I can save it as a note. So now you can see I've reformatted that data from unstructured text into a nice table. I can do something computational with it. And I can do similar things with descriptions. So I've got a big description of a species here. I'll just clear the chat window there so it's a bit easier to read. I'll send that to the language model. It's ready for me to ask it to do something. Now I can say trait table. And I get a huge list of traits back. So if I go back to my notes. Yeah, I guess everyone has been here all week, so if you recall the keynote that opened the conference, I really liked one of the things that Sarah Beery said about thinking about what is our ideal point for interaction. So I think a system like this gives researchers who are really expert in the data that they're working with access to really advanced technology so that they can experiment with it to the point at which it doesn't work for them anymore. And they can choose that point as the point when they talk to a technical team, whether that's within their own organization or an AI research team. They can do their experimenting and then have quite an informed conversation about what they need. And I can demonstrate that in that you've got like a console view of the app as well. So I've set it up to make a detailed log of how it interacted with that large language model service. So if they say, well, it got me so far, but no further, then armed with this data, they can go and talk to a technical person and have an informed conversation about it. There's a ton of other things you can do. You don't, obviously, this is a very generic tool. You don't have to use it for specimens. We're kind of chatting with some people that are active in the Biodiversity Heritage Library project about whether we could use this kind of thing to reformat some of the very structured data that we know is within those pages. And although we developed it primarily for people doing taxonomic research, there's been a couple of uses in different areas. So our colleagues at Edinburgh used it for a curation project. So they recurated camellia, and they looked at the specimens around in many different herbaria using this tool and worked out where they needed redeterminations to be applied. And also this week we heard about its use in a floristics project. So Terry Young from Paris talked about the flora of Cambodia, and she's exploring this as a way that she's arranging her project there. So it's I would say as a technologist, it's really nice to see people using the data, using a tool without me kind of like twisting their arm and forcing them to use it and saying it's part of their job. Certainly, like the project at Edinburgh, I didn't hear about it until right at the end. They just said we were productive in it, and we were happy about how it worked. We had a few ideas for changes, but they spent 3 months working on it without me kind of forcing them to do that at all. And that's testament to the amount of resources in the community that's there for this kind of thing. So if you are interested, there's a few links there. There's a website with a blog, so a few notes and so on are available. Everything that we do code-wise is on GitHub. There's a discussion board on GitHub too, if you've got ideas about different ways to use it. If you want a one-to-one demo, I'm here for the rest of the week. And we presented this also at the International Botanical Congress, so that QR code will take you to a couple of videos. Yeah, thank you.
Thank you, Nikki. Do we have any questions? I know we have some comments online, but I'd like to address any questions in the room. Erica, were there some comments or questions to add?
Just a comment on Zoom for you, Nikki, that Andre says, thanks, Nikki, +1 for Obsidian. Great tool that deserves more visibility in the Tdwg GBIF community. And then I have a question for the data you're accessing, all the stuff under remote data that you demoed. Yep. Are those static copies? Or where are you getting that data?
So it's hitting an API and it's creating a local copy. But okay, because you've got front matter there, you can always go back and repeat that communication. You can refresh it. Yeah.
Any more questions? Thank you, Nikki, for your presentation. Okay, that concludes our demonstration phase. So thank you again to all the presenters who took the risky road and did live demonstration with all the risks that entails. So thank you again. We are going to enter a discussion mode, and it's going to be pretty free form, but you're free to go now if you want to get on and go to the next event or have a break. But and you're welcome as we start to come and go as you please. But thank you, team. That's the last presentation we have on the screen, so we're just going to be discussing live in the room. I think we're going to continue to try to broadcast this through Zoom, so that will be available for people to participate by Zoom. So there may be some questions or conversation to come up there. So Deb has left, and she is our engine for throwing out idea bombs. So I don't know really where to start, but I do want to say that I continue to enjoy the outcome of this format. And before Deb left, she has to go on to an excursion, but before she left, she suggested maybe talking about how this format might be extended beyond the Spnhc or Tdwg venue, and how we might extend it maybe to some online sessions where we can just put our heads together, share our work progress, share ideas. So if anyone has any ideas or has an experience in some other area that it has done this successfully in some other venue or specialty. I mean, I think we're all very used to using Google Meet or Zoom these days, so maybe it's just a matter of deciding when and where we want to meet and what time works for as many people around the globe as we can accommodate. So I know one thing that there is a Slack channel for both Spnhc and Tdwg, so that might be a good place to start this discussion and continue it outside of this venue. I know a lot of people talked about AI, and a lot of things that we presented today was more on the technical side, and was produced by people who have spent a lot of time to learn a language, but now we know that these languages are becoming more accessible through AI. Has anyone used AI to generate code that actually did what they expected? Okay, I know Kit at least would probably have tried that. You probably could have written it yourself, but you wanted to see what it could do. Yeah. I mean, I can't quite wrap my head around pandas, but so I could probably figure it out, but it's easier with AI. So, anyone else? We at the Botanical Research Institute of Texas, we have experimented with AI. A new staff member who had no Python experience was able to come up with an idea. He knew AI could possibly solve it, and he's using it productively, not knowing a bit of Python. He just knows enough about how to run it. I connected him with how to connect to an API, and he's used ChatGPT to generate Python, to use the Google Vision OCR API, and also the ChatGPT API, much like Voucher Vision, but kind of on a kind of ad hoc, kind of local generative process. Yeah.
I'm not a programmer at all, and I use ChatGPT all the time to write code, mostly in Python.
Great.
And it's coming to sessions like this where I realize what could be possible. Listening to people about querying APIs, I do that a lot. But again, just going, I want to be able to do this, please write the code. Often it doesn't work, and you have to say, fix this, find out why this doesn't work. But yeah, it's an invaluable tool for writing. I use it mostly for writing code. Yeah. And yeah, I've done it. I did a 10-week programming course about 4 years ago and forgot everything. So I kind of know what might work. Yeah. So thank you for all the inspiration that comes out of these talks, because it's really helpful for people like me who don't have any of your skills.
Yeah. Gary, you had a comment?
Yeah, I'd like to add to that. I've talked to a lot about the AI projects that my students and I have been working on for data quality improvement. And just to really accentuate that point, most of the projects that we've been working on, I presented as part of an internship for a sophomore student in computer science. And while she did have Python background, I presented 3 potential projects that she could do in her 8-week internship, and she was able to accomplish all 3 of them in 8 weeks. So really lean into students and think, because the approachability for a lot of these tools, these services, these programs that are out there are really changing dynamically every day. So the sort of fundamental, like critical threshold of how to approach and use these tools, even as a non-coder, it's really, the barriers are dropping quite rapidly. So don't be afraid to get out there and experiment with it and talk to others about your results.
Yeah, absolutely. The resources are probably out there, like a tutorial about how to get started with Python and AI. But maybe one thing we could propose is just a quick introduction in how to bootstrap yourself into getting Python and AI working. This is a class that we are developing just for staff members where I work, but I think it could be something we could open up to the community. And you really only need maybe 15 minutes of hand-holding to get to the point where you can run some Python code and see it do some magic. So it's great to see that process, and maybe that's something we could help along. Yeah, if you wouldn't mind using the mic, I think there's still some people online. And if there are any comments online, we'll add those to the conversation.
So it's a question for people who are using AI and using to code to access APIs. How do you cope with, I don't know how it is, but for me, whenever I try to use ChatGPT, they often have incorrect or outdated information or suboptimal, it's based on old web basically, right? So I get users sometimes that come on GBIF at the help desk and they ask me oh, can I use, where can I find that? I'm like, where does this come from? It comes from ChatGPT's suggestion and it doesn't exist. I don't know why. And so how do you know? How do you do it?
Kit?
I don't have an answer, but I do a lot of coding and I do everything from data science to software development. And something that I've realized about ChatGPT is it has this curve of usefulness that I have encountered where on this side, you really need to know the basics and know the 6 things about program languaging. In the middle, very common problems, you want to run a PCA analysis on data that you already know how to access. It's awesome for that. But once you start getting farther and more in depth and really specific, GBIF specific or AWS specific, it only has a very small amount of knowledge on that and it cannot cope. So it has I think we think that ChatGPT is a magical wish granting code genie and you have to find that middle part and realize that there are parts on this side where you need to know the basics of how code works and how it compiles and what the syntax is, and in the middle super general things that you need help with, did I put the right amount of curly braces around this to run the API? But then when you get into really specific knowledge, I have found just from experience that it can't cope with that, and I think it's important to educate people on that. Did that answer your question at all?
No, but it's still good to know I'm not
Yeah.
the only one. To the point that at some point we my colleagues and I realized that ChatGPT was trained on that old tutorial that's out of date. So we're writing to the person who put the tutorial online, could you please update it because ChatGPT is screening it and spurring something out. And so I don't know. I mean, I feel it would be nice if there is AI and coding training for accessing biodiversity data, for example. It would be nice to have a little section on how to be critical, how to recognize when this is based on really suboptimal information, and how can you go beyond and check, make sure that it works, other than writing very weird question to help desk people who have no clue where this comes from. So, yeah.
I've been in a scenario where maybe the error was just a syntax error, but it came back with the error, and I think I could have fixed it, but instead, just to see what would happen, I copied the error, pasted it into ChatGPT, and I said, I got this error, help me fix it. And the response was, oh, I'm sorry, you're so correct. I should have said, and it corrected that error. So, I mean, it's actually, think of it as almost as a person who kind of knows what they're doing, and that might be the most effective approach. Yeah, we have a comment here.
I was just going to comment on that. My experience with ChatGPT is that it can be wrong, and if you call it on being wrong, it often tries to correct itself with different information because it understands that it may have given you the most generic general application of the answer, but if you say no, that didn't work for me, it says, oh well, you might want to try this instead. And that second option, in my experience, has worked more often. Okay, that's actually good to know. Yeah, and then just following up with that is to be as specific as you can with the question and like the type of software you're using, the type of platform you're using, because it does source all kinds of information. It may have old information, it may have new information, but it will use information that is the most sourced, which is probably older. It may have new information, but because the older information was sourced more in the past, it considers that more valid. But it's not necessarily the most valid for your current application.
Yeah. And while you come to the mic, I'll mention that you might also want to try different large language models like Claude AI. I found is very good with code.
I just want to, the other thing I've really been using ChatGPT successfully for is to, once I've worked out how to do something with ChatGPT, is say, please now write instructions for dummies of what we've just done. So I have a running piece of documentation, which is the kind of time-saving tips for using AI to do the work that we do. And basically every time I'm like, oh, now I can reformat this or do this, I say, now please write me instructions that anyone could understand on how to do what we've just done. And it's amazing. It'll write through like step-by-step instructions with really nicely worded prompts that you can then copy and paste, anyone else can copy and paste back into ChatGPT. And just emphasizing two other comments that have been made during this conference, and one is that it does sometimes get the API completely wrong for websites. So that's where it can trip up. So we've had, I can't remember which website it was, but it wasn't GBIF, it was iDigBio, it had completely the wrong API. I'm trying to remember the other thing I was going to say. Oh yeah, and the other thing I've been trying to do is, like, sometimes I use Text Sniper a lot on a Mac for OCR because I work for BHL. The OCR is really quite awful on BHL, and there's much better ones. But then I said, please rewrite how to do this for a PC, and it using text grab, and it wrote completely wrong instructions on how to use that. So you do, if you can't, the extrapolation doesn't always work, and you need to test things. So, but yeah, amazing for writing instructions, and also it can teach you things. So if you say, teach me how to do this, as if I was a 5-year-old, now teach me as if I was a 10-year-old, now teach me as if I was an 18-year-old, and now teach me as if I'm a university graduate. You can get it to kind of slowly, for people like me who are really hopeless, it can slowly like increase your skills.
And before Nikki, there's another comment, or Nikki, maybe you can go and then the next comment.
Yeah, so I had quite limited time to show there, but the API, the AI service that I was showing there in Obsidian, it's got this facility where you can make a load of different tasks, and you have access to them on the right click. So you can grab a bit of text and say, I want this reformatted, I want it turned into a tweet thread, I want it, explain it to me like I'm 5 years old, that kind of thing. So we can set, that's where perhaps it would be nice for us to share the tasks that we need done and the prompts that are good.
I'm so happy to hear that so many people are kind of having those barriers reduced to get into coding and are being able to experiment with tools that they weren't able to experiment with before. If a lot of the users that you're bringing on are completely new to coding and don't have a lot of experience with, I just what I'll call foot guns in how you can really mess up when it comes to coding things. So two of the examples that I'll give are like secret leaks. So if you're dealing with APIs, you have to have API keys. People are using GitHub for the first time and they don't really understand, once I commit this and then if I go back and delete the commit, it doesn't get rid of the secret. Or rate limiting things. If you kind of build a program that is just going to ping an API en masse as fast as your computer can, you might run into some reputational damage to your institution. I think it would be a really great thing to have a almost basic resource of like, if you getting started with coding and AI, here are the big things that you should watch out for, and here is everything else where you're kind of safe to experiment away. So the wrong string for the API, completely harmless. Like the only thing that's going to happen is your code isn't going to run. But the big main areas in terms of here are the foot guns you have to watch out for when you're doing development with AI. From a very basic understanding of coding would be super helpful for people.
Yeah, I think if you kind of highlight or illuminate the guardrails, it gives people a little more comfort in what they can do. So, were there any comments or questions online? Okay. Well, we should engage with ChatGPT and see what it has to respond. Well, we are running close to out of time. So thank you again for the conversation and attendance and attention. And thank you again to all the presenters. And thank you for our technical staff for helping us.